Okay, so I think we are ready to start. So welcome, good morning. We are going for this plenary session that we call Feast Forward, Feeding Tomorrow. We are going to have three speakers. The first speaker is Michael, and we're going to have the introduction for each of them. Michael comes from Tufts University. Then we're going to have Amit that is from Mulek Science. Go back. And then we're going to have Colleen from Cornell University. What we're doing with this plenary session, we are having three speakers that are going to share with us the progress that has been made in cellular agriculture and in cell tissue culture for cultivated meat. So without any further ado, I'm going to have now Vivian to come on the podium to introduce Michael, and then we're ready to start. Thank you.
Thanks, Piero. It's my pleasure to introduce Michael Saad, a fourth-year PhD candidate at Tufts, as Piero mentioned before. I want to give a little bit of background. So maybe 2019, Dr. Lucy Lee and I co-convened a session sort of on cultivated meat. And since then, we've sort of, in our society, have been bringing individuals in that are in that space. And this Feast Forward presentation, or plenary, sort of brings together both animal and plant groups. So last year, I had the opportunity to invite Michael to present at our fish cell culture on that cultivated meat, and he did an amazing job. So when it came time for us to come together for this Future Foods plenary, definitely a person that we wanted to have in our space once again. So with that, I'd like to introduce or welcome Michael to come on up and give us a little bit of a background on amazing things in fish cell culture and cultivated meat.
Thank you. Hi, so I'm Michael. I'm a fourth-year PhD student at Tufts University. I'm here representing my PI, David Kaplan, in the Tufts University Center for Cellular Agriculture. So my talk today will be on the foundational concepts underlying successful cell and tissue culture strategies for cultivated meat. But before I get into the talk, I wanted to briefly touch on the Tufts University Center for CellAg, or TUCA. So the mission of TUCA is to democratize the building blocks of cultivated meat, to build a pipeline of talent and expertise, and to foster discourse and enhance public trust. And at the heart of TUCA is research, and we've been fortunate to have received a grant from the USDA to establish the National Institute for Cellular Agriculture. This institute has 8 other partner universities and has 7 discrete aims, from things like consumer acceptance to flavor analysis. And in addition to our partners through NICA, or the National Institute, we also have select collaborations in industry, government, and academic. So thank you for listening to that little plug. Now we'll dive into the meat of the talk. So my research focuses on cultivated meat, which is a subset of a field called cellular agriculture. And cellular agriculture focuses on the production of agricultural products from cell cultures using a combination of biotechnology, tissue engineering, molecular biology, and synthetic biology to create and design new methods of producing fats, proteins, and tissues that would otherwise come from traditional agriculture. And so we see this field at the intersection of agriculture, bioengineering, and food science. And this field is set against the backdrop of traditional agriculture, or traditional animal agriculture. We know that animals provide lots of valuable products, such as food, medicine, materials, and chemicals. But animal production has a host of issues, with concerns over pollution and greenhouse gas emissions, resource utilization, especially high land use, health concerns with things like antimicrobial resistance, and poor animal welfare. And so we think that cellular agriculture is an alternative that can be viable, and we think about cellular agriculture as having 2 different types of products. One being acellular products, where the product that we would make or use is produced outside of a cell. And these include things like proteins, such as caseins for milk, gelatin for cosmetics, or ovalbumin for egg replacement. In addition to just food, and there are also other applications with small molecules and lipids with things like vanillin that are produced in acellular products. And then the other side are cellular products. And this is where the cell is the actual product that you would use in a material or food. And so cultivated meat typically focus on myocytes and adipocytes to make meat tissue, but there are other applications in materials, like using fibroblasts for leather or keratinocytes for fur. And this even extends to plant cell culture, where there are different materials, such as cotton, that can be produced from plant cells. And these two types of products have two distinct production modes. Acellular products looks like what you would see in a biopharma recombinant protein production process, where there's a fermentation involved. Typically, that includes giving the genetic code to build proteins or lipids to some sort of microbe. We think about those as being animal-free, so typically it's bacteria, yeast, or fungi. And those microbes then produce the product, which we purify and would use in an application. And on the other side is cellular products, which use a more traditional tissue engineering approach. And if we were to look at what a cultivated meat process could look like, this is a diagram that shows that. So at the beginning of the process are the cells, where you can source primary cells and immortalized cells. We then think about two distinct bioprocesses, one being a proliferation bioprocess to accumulate cell mass, and the other being a differentiation bioprocess to have more mature cell types. And in both of these bioprocesses, a key constraint is that because this field is trying to reduce the animal use in our systems, we can't use animal products in these bioprocesses. So typically that means no fetal bovine serum in our media. And once we have those mature cell types, we would then combine these into a product through a product assembly process. And so today what I'll be doing is giving some considerations and challenges for each of those big areas of this diagram, and then talking about some of the research in the field that has started to touch on these. So in terms of cells, this is the basis for the product. And we know that cells are important because they provide flavor, texture, and nutrition. In terms of flavor, cells produce proteins, fats, and other chemicals that affect flavor. And so typically when we think about cultivated meat, we think about muscle and fat tissue engineering. And we know from meat science that muscle actually provides a lot of the common meaty flavors that are associated with meat. And then fat provides more of the species-specific flavors. It helps you distinguish lamb from things like beef. Muscle and fat also play a really important role in texture, as well as having connective tissue that helps structure that all together. So cells organize themselves in their environments, and that affects the overall structure of the product. Muscle produces those fibers that you're used to having in steak or fish fillets, and fat has a lot of influence on the bounciness and the chewiness of the product. And that connective tissue is what brings it all together. And then in terms of nutrition, I think it's obvious, but cells provide a lot of macronutrients and micronutrients, such as iron and calcium. And so, as I mentioned, this field focuses mainly on muscle and fat tissue engineering. And when our lab started to get into this field, there weren't many cell lines available from food-relevant species. So we made our own. Right here I have examples from Andrew Stout, who isolated bovine muscle cells, bovine satellite cells, and has used those for tissue engineering, as well as an example from a former PhD student, John Yoon, who isolated porcine dedifferentiated fat cells and has used that in tissue engineering applications. And both of these methods are relatively simple, the muscle being a preplate technique, the fat being a dedifferentiation technique. But there are others in the field who are looking towards more technologically advanced approaches. One example comes from this company called Mosa Meat. They're a company based in the Netherlands who works a lot on muscle and fat tissue engineering, and they use single-cell RNA sequencing to inform a lot of their isolation strategies. So what they did is they performed an isolation, and then from that isolation, sent those cells out for single-cell sequencing, and they were able to identify a number of different cell types in their isolation. I'll highlight here two cell types. In blue, there are FAPs, or fibroadipogenic progenitors, and in orange are the SCs, or the satellite cells. So these are, the FAPs are used for fat tissue engineering, and the SCs are used for muscle tissue engineering, all from one isolation. And so they looked at the surface expression of different markers, and were able to then use fluorescence-assisted cell sorting in order to purify those FAPs and SCs, which is shown at the bottom right there. And once you have the right cells, another important aspect to this field is having cell lines that you can use, and that often involves immortalization. And we think that immortalization is going to be really crucial for a consistent bioprocess. And there are a number of different methods that researchers are using to do immortalization. The one on the left shows genetic engineering of bovine satellite cells. So this, again, comes from Andrew Stout, who had worked on isolating those muscle cells. And he took a genetic engineering approach. And so when he thought about why cells stop dividing and why they start senescing, he looked at the literature and saw that telomere shortening has a massive impact on that. And so telomeres are these caps on the end of your chromosomes that keeps the chromosomal information intact. And as cells divide, those telomeres get shorter and shorter. And eventually they become so short that your DNA shears and you get senescence. And so what Andrew did is he overexpressed a telomerase reverse transcriptase enzyme as well as a cyclin-dependent kinase 4. So that telomerase reverse transcriptase helps ensure that those telomeres are quite long and allows the cells to proliferate, hopefully past the Hayflick limit, which is the number of doublings where you start to get that senescent event. And then the cyclin-dependent kinase 4 is a cell cycle progression marker, so getting the cell cycle to move from G1 to S. And his data showed that his hypothesis worked, where in pink we have those engineered cells, and in black we have the non-engineered cells. And you can see that after about 50 days in culture, that black line starts to plateau and the cells actually had died, whereas his engineered cells, the one that's in pink, kept growing indefinitely, and they've now been in culture for over 200 passages. And while genetic engineering is one approach that people are using, spontaneous immortalization, just through random events, is also another approach. And so the figure on the right comes from some work that I had done on fish muscle. So we isolated fish muscle cells from the Atlantic mackerel, and we had noticed that our primary cells grew, but eventually reached some crisis event where we saw high rates of senescence in our dish. And that's visualized here by those arrows pointing to senescent cells. But you can also see in that image that there are small pockets of cells that look healthy. And so what we did is we just continued passaging these cells, continued to grow them, were patient, and eventually those healthy cells started to overtake our culture and have now been in culture for 2 years. And importantly to us, we look at the doubling time. Our pre-immortalized cells grew at about 60-hour doubling times, but now our cell line grows quite well at 24-hour doubling times. And while we think a lot of these approaches are great, there's also a need to think about how to scale up cell lines. And we also think that an important consideration here will be adaptation to suspension, to have adhesion-independent growth. And so we have a couple different approaches in our lab that we're using. One comes from Sophie Letcher, who works on insect cells. If you're interested in hearing more about her work, she presents tomorrow at 10:30. But what she had done was take an insect cell, which again is maybe a non-traditional type of food cell line, but she does a great job pitching the cell line. She took insect cells and isolated them from caterpillar embryos. She could then adapt her cells to serum-free media quite easily, and then had noticed that there were some cells that were just naturally peeling off the plate. Through a selection process of those non-adherent cells, she was able to continually purify those cells, and eventually has a non-adherent cell line, which she has termed the Manduca sexta non-adherent cell line, or MSNACs. And that cell line is great, and on the other side of things is a chicken embryonic fibroblast process that comes from a company called Believer Meat. So they isolate chicken embryonic fibroblasts, which are adherent, and after an immortalization and serum-free media replacement, worked on getting those to suspension. So their process was to first encourage aggregates by seeding these fibroblasts into agrowells. Those aggregates formed really nicely, and then over the course of a couple months, they started to gradually increase the shear in which they grew these aggregates. And eventually those aggregates started to dissociate into smaller aggregates and even into single-cell suspension. And we know that this process works because our lab has actually used their protocol in order to take commercial DF1 chicken fibroblast cell line that now grows in suspension in our lab. And so that's my considerations for the cell. And now I'll touch on the proliferation bioprocess. And I think when I first started in this field, when I thought about the way to make this most viable, I thought that the growth rate was going to be the most important. Can we get these cells to grow fast to make this efficient? But in talking to companies, they often think that the cell density might even be more important. So the cell density, if we can increase the cell density and lower the space that's used, that would mean less bioreactor runs and less CapEx associated with that. And in both of the cell density and growth rate considerations, we need to think about the media and how that affects cell metabolism. So in order to get cost-competitive products, we need a low-cost media that works efficiently. And as I mentioned, an additional constraint here is that we need to remove animal products from our media. And in removing animal products, that former PhD student, Andrew Stout, took a project on to adapt his bovine cells to serum-free media. So when he did this, he looked towards other literature and found that there was an iPSC B8 media derived by Paul Burge's lab at Northwestern, and he tested those on his bovine satellite cells. He saw that those cells didn't grow just in B8. They didn't die, but they didn't grow. So then he started to explore what he could add to B8 in order to get the cells to grow. And as you can see in this figure on the left, he added recombinant human albumin and saw that as he increased the concentration of his albumin, his cells started to grow more and more. And that was a short-term assay. He then tested this in long-term, as is seen on the right, So in blue here is that B8 media alone, and you can see that after 15 days, the cells aren't really growing and start to die, whereas the green and pink bars show his BF9 media at 2 different FGF concentrations. And so you can see that over 30 days, he was able to get nice, consistent growth of these bovine satellite cells in serum-free media. They still don't do as well as that black bar. That's his BSC growth medium, which actually contains 20% FBS, but that just means that progress still needs to be made. So he was really excited to see a serum-free media. That recombinant albumin was in quite high concentrations and was leading to quite high costs. And so he explored different ways to replace that recombinant albumin, and he looked towards plants. So he tested a number of plant protein isolates. The one shown here was the one that worked best for him, a rapeseed protein isolate, which comes as a byproduct of canola oil processing. And he was able to show that when he added this rapeseed protein isolate at a concentration of about 0.4 mg per mil, he was able to get equivalent short-term growth to that BF9 bar, which is on the right side of that left graph. He then wanted to confirm that this worked in long-term culture, and so he performed long-term passage-to-passage assays and saw that purple squares over there, that BFR media, did better than his BF9 control, which is the previous media that contained albumin. And then he, again, wanted to see ways to make this even cheaper, and him, having had done the immortalization of his bovine satellite cells, knew how to genetically engineer these cells quite well. And so when he was looking at the cost of his new media, he noticed that FGF, fibroblast growth factor 2, was in quite high concentrations and leading to quite high cost of his media. So he was interested in actually producing an autocrine signaling cell, so a self-signaling cell that would produce its own FGF and then reduce the need for FGF in the media. So what he did is he produced a number of different constructs. He produced an overexpression of FGF, as well as an overexpression of a mutant RAS, a G12K RAS, or G12V RAS. And that RAS is constitutively active and a downstream effector of that FGF binding to FGFR. And so he made these different constructs and tested them in his growth curves shown on the right. So his controls here are those blue circles, which is his positive control. It's the wild type cells grown with FGF in the media. And the other control is the negative control, that purple square bar that just doesn't grow. So that's his bovine wild type cells grown without any FGF in the media. But you can see all the rest of the lines are his engineered cells, and they all grow quite well. And I'm going to highlight his top 2 growers. So that are the 2 lines that contain the yellow in them. And so that was his double knock-in of FGF overexpression, as well as the constitutively active RAS. And that bar that even did the best was his BFR media, the rapeseed protein media, growing these engineered cells. And so while media has been progressing, and there still needs to be improvements to catch up to as well as FBS does, the next step in this field is really thinking about how to scale up these bioprocesses. And when we think about scale up, we often think about different bioreactors to support cell growth and differentiation. And so the most common bioreactor that you would see at companies are these stirred tank bioreactors that use an impeller to provide shear to the cells, quite common in biopharma. There are others in the field that are looking to replace that impeller in gas in an airlift bioreactor to have gas bubbles do a lot of the mixing. And there are others in the field that are working on more anchorage-dependent methods and looking at how hollow fiber reactors can work. And they're even producing hollow fibers that are edible. And so you would have a hollow fiber system that you'd put into a bioreactor and would have a perfusion system feeding that. But in this field, scale-up is the hot topic because it's going to be hard. There needs to be innovation and infrastructure to make this feasible. And so I'm going to highlight the need for infrastructure by really quickly going through a short thought experiment. So by 2050, it's estimated that the global consumption of meat is going to be about 500 million tons. And while we think this field can do well, we don't think we're going to displace that much production very quickly. So if we assume that we can reach 0.2% of that, that would mean we need to produce 1 million tons of meat. And based on current estimates, the volume or amount of bioreactor space that's needed to achieve this would be 1.5 to 3 billion liters. That's a ton of space. And when you think about it in terms of Olympic-sized swimming pools, that's about 750 to 1,500 Olympic-sized swimming pools worth of bioreactor space. But our current capacity right now is just 10 to 20 million liters. So all of biopharma, if we were to fill those reactors up, it would only be about 10 pools. And as you can see here, that's not very much when you consider the 1,000 that we'll need to make this meat. But that just highlights the need for innovation. But we don't only need to innovate and have public infrastructure that helps us scale this up. There are also innovation that needs to happen to make these products cheaper. So the figure on the right here comes from David Humbird, who performed a techno-economic analysis of cultivated meat. And he found that even at really high production volumes, so on the very right of his figure, even at really high production volumes, the cost of cell mass with his assumptions would be about $40 per kilogram. So definitely more than you would be used to spending at the grocery store. And that just highlights the need for innovation. And so when we think about innovation, we think about ways to think beyond constraints of traditional biopharma or tissue engineering. I think Andrew's example of self-signaling cells is a great example of that, but we need to think more. And so when we talk to people in industry, some of them have the mindset that we should just start to attempt scale now, see what happens, see what failures are made, and learn from those failures. Others look more towards Andrew's kind of approach in genetic engineering and how that can help make these bioprocesses cheaper. And even others are looking towards more simple processes and looking towards nature to do this. And so when we think about what cells would be ready to scale up, I think the suspension cells that I mentioned earlier are the ones that we have that are the most relevant. And so Sophie's M-SNAC cells can grow in suspension, and in a relatively simple process of fed batch, she's able to get about 30 million cells per mil of cell density. Without media exchange, she can get 4 million cells, and she's exploring ways to make this even higher, so definitely recommend going to her talk tomorrow. And that Believer Meat paper that used the chicken embryonic fibroblasts, in a fed batch process, they can get about 7 to 8 million cells per milliliter, but they also have a really impressive perfusion process where they can up that cell density to about 100 million cells per mil. And that, when you think about weight to volume, led to about 36% weight of cells per volume in their bioreactors. And I mentioned that scaling these processes is great, but those fibroblasts that I mentioned aren't the muscle and fat cells that you're used to eating. And so in order to get the best of a scalable process, as well as that phenotype that you're associated with, there are groups that are looking towards genetic engineering. And one example comes from a company in the Netherlands called Mutable. Mutable licensed a technology called OptiOx for induced pluripotent stem cells that would then have inducible muscle and fat generation, so inducible transcription factor engineering. And so in that paper, they show that they could isolate iPSCs from humans, and Mutable uses pork cells, but they could isolate iPSCs and get them to grow in aggregates. And then by adding in inducers for different transcription factors like MyoD, or the myogenic determination protein, that paper was able to show really nice muscle differentiation shown by their myogenin and myosin heavy chain positive staining. And then in terms of fat, Mutable is looking at PPR gamma in order to get fat aggregates as well. And I can't share some of their images, but Sophie and I have had the pleasure of going there and seeing their images. They have really cool differentiation processes where they can grow these aggregates and actually get those aggregates themselves to differentiate. And so you can see these like massive aggregates of muscle and fat little spheres in their images. And while that process is really cool, genetic engineering is still kind of questionable as to whether it'll be accepted, especially in Europe. And so others are looking towards more natural materials to help us scale. One example comes from Glenn Gaudet's lab at Boston College. And he focuses a lot on decellularization in order to make scaffolds. And so one of his students, Jordan Jones, who's now a postdoc in our lab at Tucka, thought that when he was looking at a spinach, it had really nice natural veins that he could use. And so he was interested in using spinach as a scaffold. The video is not playing, but his paper is great, but his most famous claim to fame is a TikTok that he made that had hundreds of thousands of views on TikTok, showing the decellularization process of a spinach. Another person in Glenn Gaudet's lab, Rick Thyden, also wanted to use decellularization and noticed that the little broccoli florets that he was eating actually looked like microcarriers that cells attach and grow on. And so he made edible microcarriers by taking those broccoli florets, decellularizing them, and putting bovine satellite cells on them, and showing that they could grow in a bioprocess. And while it's great to have cell mass through a proliferation bioprocess, in order to make these products most close to recapitulating the products that we're used to eating, we need some sort of differentiation and product formation. So as I mentioned many times, muscle and fat differentiation are going to be pretty important, and they impact flavor. And then in terms of product assembly, there are a couple different methods being explored. One is whole cut tissue, so making 3D tissues, but others are actually using cells as an ingredient. And so in terms of groups that are looking at how to improve the differentiation of these cells, Mosa Meat, that one startup in the Netherlands, is pretty firm on requiring that their product will have muscle and fat in it. And so they've been identifying ways to make better muscle and fat media. What they've done in the study shown on the left is they did a RNA-seq study where they analyzed the receptors that were upregulated during their traditional serum starvation, muscle differentiation. They looked at those receptors and identified ligands to those receptors that they could then add in a serum-free media and made a serum-free differentiation media. So you can see on the images they have that their serum-free media does quite well when compared to their serum-starved media. And on the fat side, they're also looking at ways to make the fat differentiation more simple. So typically, an adipogenic differentiation has a two-stage process of induction and progression, where you add agonists to PPR gamma to get the cells to accumulate lipids. And what most of meat had done is they actually thought about ways to simplify this. They looked at removing certain components and have a reduced differentiation strategy where they just add two components, rosiglitazone and insulin, and term this their reduced differentiation media. So you can see in the images, both with serum and in their serum-free DMAD, that the reduced media actually outperforms that traditional media that most people use to differentiate into adipocytes. And once you would have those differentiated cells, you would then need to figure out how to get them into a product. And we do think that cell differentiation can improve the organoleptic properties of those products. And so the figure on the left here comes from John, who worked on that pork fat cell line. And what he had done is he had looked at ways to get a 3D tissue from fat. And he would take his cells, grow them in 2D, have them accumulate lipids, and then scrape them. And he would then mix those scraped cells in an aggregate process with a different biomaterial. So over here is shown an alginate. On the left, that clear material is just the alginate. And on the right, that nice looking piece of fat, that's his alginate fat mix. He was able to produce enough that he could actually do texture analyses. And so we performed DMA, to get the elastic modulus of our his different fat products. And he had seen that when he had the alginate with his fat, that led to a more similar elastic modulus to pork fat, as opposed to him just having alginate. And so in terms of texture, we think there could be improvements. And then when we think about flavor, like I mentioned earlier, muscle often provides that meaty flavor, but it hadn't really been shown in the literature too much, at least for in vitro processes. And so there's a group out of Korea that produced a type of muscle tissue there, or muscle and fat tissue, where they would have different layers of muscle and fat, and they would then take their muscle and fat layers and perform flavor analysis through GCMS. And so the graph over here shows some of that results of their volatile analysis, and the HALC, that's the one I want to focus on, that is their muscle cells on a scaffold. And you can see when we look at the roasted beef flavor in the very right, the only time that we can detect that they detected that roasted beef flavor was in their scaffolds with the muscle cells present. And as I mentioned, different groups are taking different approaches in order to forming products. So both ground and whole cut products are being attempted in companies. The figure on the left here shows that Believer Meat chicken fibroblast paper, and they would actually do a lipid loading process, so it wasn't a proper adipogenesis, but they're able to get their cells to accumulate lipids. They're feeding with lipids, and they can then use those lipid-loaded cells as an ingredient in food products, and you can see an example of their cell paste there. On the other end are groups who are thinking more towards proper tissue engineering, and there's a group out of China looking at porcine skeletal muscle. And so what they had done is they took these molds that are little PDMS molds that create these little chambers for nice muscle differentiation. They would load those molds with their muscle cells, allow them to mature into muscle fibers, and then they would aggregate those muscle fibers together to make products like their meatball shown in the bottom right. And while a lot of this research that I've shown is what's publicly available, a lot of the funding in our field has gone towards private companies to produce products. And so there have been some success at the private company level. One example is from Upside Foods, a startup based in Berkeley, who had partnered with Dominique Crenn, a really well-known chef in San Francisco, to produce a really fancy-looking fried chicken dish. There's also a company based in San Francisco called Good Meat, who partnered with Jose Andres in Washington, D.C., to make a Peruvian chicken skewer that they served at one of his restaurants. And a company in Australia called Vow looks towards more non-traditional sources of meat. And they just released their first product in Singapore, which is a cultivated quail product that you can purchase at a restaurant there. And while I think it's great that we see some example of this technology being adopted, I do think that there's a need for more open collaborative research. As I think I've laid out in this presentation, there are really quite challenging problems that the field needs to solve in terms of media, in terms of scaling, in terms of bioprocess. And the best way to solve that, in my opinion, would be through open access, multidisciplinary research. In addition, there's going to be a need for more public infrastructure, or at the very least, industry-academia partnerships in order to hit some of these goals that we hope in displacing traditional animal agriculture. I think when we often think about a field that has scaled well, has scaled cooperatively, one that often gets mentioned is the semiconductor industry and how they came together to achieve Moore's Law and scaling over the last 30 years. And so our institution, TUCA, the Tufts University Center for Cellular Agriculture, is just one example of a hub in this field that is open to collaborating with more researchers. So if you're interested, please come find me or email TUCA, and we'd be happy to explore collaborations. So I want to thank SIVB for the opportunity to speak here, and I want to thank you all for listening, and I'd be happy to answer any questions.
So I have a provocative question for you. So has to do with media, but other kind of media. I could easily see the headline, Company X selling us GMO cancer cells. Have you thought, how are you really getting ahead of the messaging? The messaging on GMO plants, we made a stupid mistake. We were not ahead of that messaging. And now even maybe in this room, there's some people that think, oh, I buy the non-GMO stuff. How are you getting ahead of that?
Yeah.
And if it's a totally inappropriate question,
No.
just tell me to sit down.
It's a question we get a lot. So actually, after the first products were approved for regulatory approval in the US, a lot of publicists or a lot of media reached out to our PI, David, with that exact question. How do you know these cells aren't cancerous? That has led to us now studying genetic stability in these cells as a way to hopefully show that we have a stable cell line that's not going to mutate too much over a bioprocess. But there's also the argument to be made that when you eat these cells, they're going to be degraded in your body. They're not going to be viable anymore. And so that's an argument that the companies are making right now. But they're also doing safety testing. They're doing RNA-seq analysis over multiple passages. They're doing proteomics to look for allergies. And so there's safety dossiers by the FDA that are now being made more available. I don't have the great answer, but that's some of the considerations that we're thinking about.
We have another question just over here.
Thank you. Nice talk, Michael. A comment, one other thing to worry about, and that is the livestock industry and how they're going to, deal with this. I know that there's already been concerns of that when David was working on other proposals. But what's the weight volume of meat in general? Because you gave some numbers, and I was curious what the weight volume is. I don't know. So if you look at, say beef or chicken, because you were throwing some numbers out of 36%. Is that close?
I think that the weight volume that I provided there was a wet cell weight.
Yeah.
And so I think that would be equivalent to like 100% of a traditional like chicken or beef product.
Oh, so that matches it closely then. Is it what you're saying?
I think if you were to take those cells, to wash them a few times, and then to use that cell paste, I think it would match somewhat closely. I haven't done that analysis. I think others in the lab have. And I think there are some differences, but they're pretty small.
Thank you.
I have a simple practical question. So what would be a cost of a hamburger made from single cells? Is it price competitive, and what would it take to make it competitive?
Yeah, so again, we work in academia, so we don't know about the cost of these products. My assumption is they're selling at a loss right now, so I think it is still pretty expensive. The first time that this technology was really demonstrated to the public was in 2013 by Mark Post, now the founder of Mosa Meat. And I think his 2 hamburgers cost $500,000 in that first iteration. But he says that their cost has gone down, and I think if I had to guess, we're probably at $100 per kilogram in the companies, but again, it's just a conjecture on my end.
I don't think I have a good question, but I will try to make my point. You were projecting that 0.2% of meat production, about 1 million tons, right?
Yeah.
Let me spin the question like this. I mean, this is definitely something new of interest. Have you thought about producing meat of pharmaceutical value? You know?
Oh, like nutritionally enhanced meat, or?
Sort of. Like
Yeah.
even like vaccine type, you know? That could be of something.
So that is a project that we're actually doing in the lab. So we have looked at vaccine meat and engineering the bovine cells to express antigens to different diseases. And we've also looked at ways to nutritionally enhance the product to make it even more healthy or tasty than typical meat that you're used to eating. So we've worked a lot on fatty acid desaturase to increase the omega-3 content in some of the product and some of the cells that we're producing. But yeah, so we have had more like biopharma type applications, but we're also looking at making even enhanced products.
We got time for one more?
Excellent presentation. Thank you very much. I just have two questions. The first was, early in your presentation, you had shown that you had naturally selected for proliferating cells in mackerel, and you'd gone 200 passages, and I was curious if you had a chance to analyze them genomically or translationally and compare it back to the source tissue. And my second question was, did I hear right, but the iPSC cells were derived from human in the Netherlands?
I'll clarify that one first. So in the paper that Meatable used, where they licensed that OptiOx technology, that paper was in human cells, but they use pork cells. So they have pork iPSCs. So sorry about the confusion there. In terms of looking at the immortalization pre and post crisis event, that's something that I'm actually actively doing research on now. So I've done RNA sequencing, and we've done whole genome sequencing. And hopefully by this time next year, I can present some of those results.
Great, wonderful. Thank you so much, Michael, for an excellent presentation.
Okay, so now we're moving to this next talk, and we're moving now into the talk from Amit Dhingra. He's the Chief Science Officer of Mulec. He has 25 years plus of experience in academia, and he's going to switch now into molecular farming for the production of animal protein. So, thank you, Amit.
Good morning, everyone. I hope you can hear me. Yeah? All right, just want to make sure everybody's awake. So after, that was a great presentation, very heavy on science. This will not be heavy on science. First of all, a few acknowledgements, and I am just acknowledgement of a conflict of interest with my position at the academic institution. So there's a conflict of interest management plan at A&M. Just wanted to get that out of the way. Today I'm representing only Mulec Science here. So molecular farming. I know that there are several leaders, people who really founded this field back in the 80s. And so this is true, tried, tested methodology. And it's just great to see a lot of friends, colleagues, and my mentors here as well. So I'm a bit nervous, right? I've got my bosses here. Another disclaimer, since Mulec is a... All right, you don't have to read it. I'll summarize it for you. This is not a solicitation of anything, any sorts. Just these are forward-looking statements, and so what I can present is only what has been publicly disclosed. So just bear with me, and if you have any questions, of course, we'll see what we can do with that. A great team really makes things happen. So this is not really a story of science per se. It's a story of people coming together and making things happen. And I know that several of us in academia and people who are working in industry are dedicated to getting the products to market. I've been exposed to several such efforts throughout my career as well, and I've seen them fail as well. A great team really takes it all the way. So I just wanted to acknowledge that. But there's another story over here. Gaston Palladini is a fourth-generation pork producer and one of the largest pork producers in Argentina. Anybody here from Argentina? All right, you probably know the Palladini last name. Yeah, see, I was not lying. And then so Gaston and Hank and Martin, these guys got together initially. This was during the pandemic, when they really wanted to express some of these animal proteins. And of course, that's the time when alternative proteins were becoming big. But they were really interested in coming together and starting a company. And Gaston was really inspired by the fact that he wanted to bring some sustainability because he was producing so much meat. In fact, he jokes that he grew up in a slaughterhouse because that's where he used to be as a child. But the point is that we've really come together as a team, a very diverse, multidisciplinary team that is really moving this forward. I want to acknowledge Vivek and Bruce, who are our senior scientists in plant biology and also in fermentation science. We're really pushing the frontiers. So none of you are unfamiliar with the fact that we have all these challenges going on. The strife around the world is creating food challenges, food supply challenges. Environmentally, we are facing extremes of climatic conditions all the time. And nutrition. This has been a perennial problem. I grew up in India in the 70s. I know there are other folks from other parts of the world where nutrition remains an issue. I personally experienced that. My mother used to be a medical doctor, and I used to see her trying to supplement the nutrition of poor women who could not, poor financially, women who could not really have access to nutrition, and thereby they had challenges. So there are just lots of issues that are ongoing. And finally, this slide talks about diseases as an animal, but we also know that there are, of course, pest and disease issues with everything out there. So this context is very critical, why this Mulek science exists as well. Again, you don't have to read this, but just an overview. We are using soybean as one of our key platforms, and that's because it's I think over 90% of soybean grown, at least in the United States, or even more, is GMO, as well as in Argentina as well. And then it's 70% of these seeds. I did not know this. I learned this from Gaston after I joined Mulek. 70% of the seeds that we do produce are fed to livestock, and that becomes our food. So there is a lot of effort going into produce food for the food that we finally end up eating, which is the animals, of course. The food to feed conversion or feed to food conversion factor is also very inefficient if you look at the animal side. And of course, over 40% of greenhouse gas emissions can be attributed to this industry. Again, we are growing. We are always, the population is really exploding. How are we going to support that? So you just heard a great example previously for the cellular agriculture process. So this is another approach that we're taking within Mulek science. So traditionally, we source proteins from animals, but also plant-based proteins. In India, for example, a lot of legumes are utilized as a source of protein for vegetarian diets as well. But that kind of, in the United States context, as you must have seen, the Impossible Burger and all these plant-derived proteins became a major source, and there was a big, momentum towards that. But it kind of stalled, and the reason is taste, flavor. Of course, price parity is another issue, but the flavor really killed it. And a lot of consumer studies have shown that our human behavior is guided by by flavor. If you don't trust me, go ahead, and that's why the golden delicious apple or the red delicious apple faltered, because they did not provide you consistent experience. I take an example of a fruit because I work on fruits as well. But just this is commonplace. If you don't enjoy the flavor, you will not come back to that point. So there's a big push for that. Fermentation is another platform from which a lot of the protein is now being produced as well. And then finally, you just heard about the cultured meat, so I will not even attempt to elaborate on that one. So the idea is that Mulek decided they're going to be like a category creator in this process, where they're going to be able to meet with taste and texture, and I'll explain how, nutritional value by engineering these animal proteins in soybean. And then, of course, there is a big, huge infrastructure already existing around soybean, all the way from growing to crushing to processing and adding it to food, utilizing it as a food ingredient. So the entire pipeline already exists in that process. This Good Food Institute also did an opinion piece on this, that plant molecular farming is emerging as the fourth category. When I show these slides, I always think, but I started doing molecular farming in the 90s myself. So it's not new, but I think the idea of expressing animal proteins as food ingredients is new. That's the novelty of all of this. So Moolec is basically a food ingredient company focused on the use of molecular farming technology, and we really want to create food ingredients by engineering plants with animal protein genes. It's a spin-off from a previously publicly traded company called Bioceres. They had initially, their proof of concept was the chymosin protein. It is the enzyme that is utilized for production of cheese. So they had already gone through the entire process, and in 2019, 2020, they spun out everything. So, and I actually joined them in fall 2020. While we were all glued to our Zoom cameras and our computers, that's like, what's one more thing to do in life, right? So this slide really sums it up. Today, 70% of the seeds we produced, we feed that to livestock, and then we basically make food. And the idea is that we start utilizing protein genes that are important for organoleptic profile, basically flavor, texture, aromas. Those proteins can be now engineered directly into plants, and the way we are using is we're using seed-specific expression because soybean is the seed is the key ingredient, and then that product can be utilized as a food ingredient. These are some ideas why this is cost-effective. Of course, there was a question about cost as well. Plants and bioreactors, several of us who are plant biologists here know that. We really don't need to worry about putting big infrastructure around those. No extra purification cost. You can utilize these proteins, the flour. So soybean is fractionated into oil, protein, and carbohydrate, starches. So that fractionation already exists. So you can just plug it into that same system and process further. And the other part is that this can be truly scaled globally because soybean are grown everywhere in the world. This can, and by breeding this trait into local varieties, you can really scale it globally. And the big part is you can involve farmers in this as well. So value add for the farmers as well. These are some numbers that you use compared to animal rearing. Land usage is reduced, so is water footprint is reduced, as well as CO2 emissions can be reduced. Now, the idea is not to completely replace the animal industry. It's just as another tool. And you'll see there's a very specific application where this comes in handy. So now I'm going to get a little bit of overview of the product pipeline. Today, meat replacements are already in the market that Moolec is producing. Moolec has a crushing facility, processing facility, where they are processing soybean, regular soybean. So that is already in the market, that pipeline already exists. Second part is that chymosin and gamma-linolenic acid, which is a nutritional oil. The safflower is the platform there, and these are all both patented. In fact, the nutritional oil is getting into the market soon, as well as the chymosin piece. The final part on your right would be the three products. One is based out of fermentation. It's an iron supplement. And then, of course, we are here to listen about soybean, which I'll talk about shortly. Soybean, actually, we developed these lines. I'll give you an overview in partnership with the Wisconsin Crop Innovation Center. Hi, Albert. As well, the pea product was developed in my academic lab through a sponsored research project when I was at Washington State University. So Glasso is the gamma-linolenic acid. This is already being scaled up as we speak, and it's in the process of commercialization. GLA is found naturally in several crops, of course, evening primrose and borage seeds, but in Moolec safflower, through this engineering, there's about 50 to 60% GLA, which will get utilized in various applications from humans as well as animal feed. So we received the RSR for this particular product in 2023. So this product can be produced and moved forward as well. So this is the soy pipeline, which we worked with the Wisconsin Crop Innovation Center. We had several, so my background is in regulation of gene expression. And so we had developed specific constructs to have high-level expression in seeds, in soybean seeds. Just going through the step, we had about 550 T0 events. We've been narrowing it down by looking at specific key performance indices, which are stability, copy number, expression levels, and now currently we are at T4 generation. So we've kind of slowly, it's like a funnel, started with a large number of events because we, at the end of the day, want to select an event that's highly stable in different agronomic conditions and also make sure that the protein levels remain high. So we were able to get 26% total soluble protein in these seeds. That pink hue is the source of inspiration to call it piggy soy because it, it looks cute, right? You guys have gone silent, come on. All right. So we've actually not, of course, we are in the process of tasting and trying these things out. So when you try to cook them, it does, obviously there is iron, so the protein is myoglobin, which is the key protein that binds iron. And that's what really gives the flavor, the meaty flavor to the meat. So this is really cool. We don't know how much iron is bound to the myoglobin, but those are some studies we are currently working on. So it's pretty cool to see this come through. And of course, we have several lines that I just showed you in the past slide. We have about 140 T2 lines. We've selected 10 top lines in this case, and they range, everything is above 10% TSP. So just comparing head-to-head with the regular soybean, there's not much change in the composition. There is a similar amount of protein, oil, fat, moisture, and minerals in there, just that there is 10% porcine protein out of the total 35% protein. So this is a value-added product that can really enrich the food as well. Now, you must be wondering, what's the use for this? And I'll just tell you shortly. So I did not know this. I don't know if you knew this, but our processed meat, nuggets, burgers, patties, it contains something called as texturized vegetable protein, 5 to 50%, depending on the quality, and of course the price as well, because you can easily replace meat by adding TVP and reduce the price because it's soybean protein. Now, soybean protein on its own doesn't have all the flavors. So now I hope it really connects the dots that you can use the piggy soy TVP and add it to these products. So you're adding nutrition along with flavor into these products as well. So that's right there, that's the ingredient part as well. So there is so much unnatural chemicals that go into producing this type of protein, or this type of products. But this is just the first product that we have moved forward with. But you can say we can't replace everything again. This is just another toolbox that we have. We can start slowly making these things, keep the price low, increase the flavor, increase the nutrition. That's the goal. And just in April of this year, MULAG became the first molecular farming company to achieve this USDA approval. So we got an RSR for piggy soy. So we can grow it now and do field trials and start collecting data for next steps. We have operations across different locations, and thanks to that crushing facility in Argentina, in orange are the current customers for the product. So you can also see the vertical integration. So as a public scientist, we always dream of bringing things from concept to people. This is a great way to show that there's a pipeline that exists from crushing to the customer already in place, so you can plug in these products in and take it to market through, of course, after you go through the regulatory approvals. Recently, in partnership with Bioceres, MULAG also acquired the HB4 platform, which is the drought-resistant soybean. Again, talking about sustainability, integrating a lot of these traits together, not just nutrition, but produce it sustainably so that farmers can make sure that they can get the crop and they can get the money back to them. So just wanted to say a vision of a single individual can go very far, and this is Gaston Palladini. He's really built a team and a concept together that not only brings people together, but partnerships together. So I also wanted to say if folks in the audience are interested in partnering, thinking of new ideas, please reach out to us. There's always room for collaborations. and partnerships as well. So thank you very much for your attention. I'll take any questions you may have.
So we have time for a few questions.
I know it was not a science talk, so.
Well, no, we've got one right over here.
Oh, all right.
Can you use this for soy milk? Could you possibly use the product?
Can we use it for soy milk? I think that should be possible because it's a protein fraction that would go in there as well. That's a great idea. Well, okay. I'm already tasting it. All right.
So 2 simple questions. First of all, you showed a picture of these modified or the soil. And if you go back to the slide, but I just described the normal one looked like yellow. The new one looked like a little bit red. Is that true color?
The question, everybody got it, is that true color? That's an absolutely true color, and it's based on the percentage TSP, all the way from 3% to 26%, we can see actually a gradient. In fact, the color starts showing just over 4% TSP. Yeah.
Very cool. So second question is, my understanding this would be for human consumption at the end. Is there any regulatory issue if you really become the food?
Yeah, of course. We have to go through the FDA approvals for this, so that's why first we have to collect all the data for that as well. Yeah, I think the first phase is APHIS, which has been done, and next phase would be that. Yeah, great questions. Thank you.
We have another one over here.
Can you tell us about the cost? Would it be cheaper to produce chymosin or piggy's protein in yeast rather than in plants?
Sure. The cost factor is being calculated for chymosin. I think that's in a public disclosure. It's at par with what you would produce through other processes. But the other idea is that to maintain a fermentation facility versus growing it through the farming process, of course, it comes with risks as well. I mean, we need all sources. It's not one or the other. I think that's one mindset, at least I want to make sure that we convey. It's not one or the other option. We need all these tools, given the situation we are in. But great question, thanks.
So looking at this from kind of a metabolic and human health perspective, we understand that animal-based proteins have the capacity to present several different comorbidities, things like cardiovascular disease, obesity. So I'm wondering if we channel it through a plant source, do we see some of those same repercussions?
That's a great question, to be determined. But I would say that they also say if you eat fruits and vegetables with your meat, it doesn't affect you. Here you go, got the package now. But that's a great question. I mean, I say it that response is in jest, but those things will be determined once we have the approvals to go forward. But we are already eating meat and soy, and the TVP is being utilized, for example. And the other part is access to protein and nutrition. Just we are thinking of people who have metabolic disorders who have access of food, or access and access to food. This could also solve problems in places where it's hard to get meat to areas where you don't have storage or cold storage chains. Well, you can grow soybean there. So that's the other angle to think of from another perspective. Yeah.
So Michael is that speaker. My question is actually very similar to that. There is a historical famous story for just consuming a lot of protein from animals. Like when Roman Empire conquering Spain, they had an issue with supply chain through the Pyrenees mountain. So basically the regime leader asked the soldiers to eat rabbits. Rabbits has huge animal protein composition, about 30%. I mean, that's one of the highest protein from animal concentration. As a result, many Roman soldiers starved to die because the energy they consume to digest those proteins is higher than they needed. They collect it from the food. So basically from this similar strategy, if you convert those plant protein to animal protein, then you put those countries, developing countries, when they are already starving for food, you might have a serious issue when you put them just like Roman soldiers.
I'm speechless. You know, I think that, as you heard in the previous talk, we are starting to develop these new products. What will be the ramifications? I think that's the area. So what you're identifying are areas for future investigations. But in the meantime, we can at least start replacing the TVP that is in those patties and burgers, at least start providing some nutrition that is available. But also, I will say, you know, you're adding some, it's not pure protein at the end of the day. You're obviously creating a product that is plant-derived as well. So, I mean, if I were to venture to say that, you know, with soy-based product, I mean, maybe there are some benefits that come from this plant-based product as well. Yeah.
For developed countries, where it's, you know, healthy.
Yeah.
But for developing countries, let's be careful.
Yeah. Well, we also have a product with peas as well, right? So, I mean, that's also consumed quite a bit around the world. So, I know that iron deficiency is one of the biggest challenges around the world, even today. So, I think adding this type of protein that supplements iron would be a good option. I'm not saying the only option again. Thank you. Yeah, yeah.
Hey, here. Just, I was thinking, like, as far as I remember, 94% of the cultivated soybean had like the herbicide tolerance, right? So, and that was an issue, but before was just like they are for animal feed. So I was thinking if you have that in here and what it's about the market acceptance on that.
Well, right now we've produced this product in the common line that is used for transformation. But once you start introgressing into different lines, it could be herbicide tolerant, it could be non-herbicide tolerant, it could be other type of, because you need to be able to grow this properly as well at high yields as well. Or maybe you can grow it organically. I mean, there are so many options post the approvals where we finally introgress this straight into. But those are some considerations. We are all thinking about those. I think the laser focus right now is to ensure that we can kind of take it as a category to the market. You can kind of see we've all been working here. I mean, I worked in many labs with colleagues over here as well. We're all trying to get this concept to market. And there was a concept earlier about cellular farming. One of the colleagues mentioned about the public perception issues as well. I mean, if we get this to market, soybean is already GMO. It's already in the market. So there is a possibility that it won't be easy, but somewhat relatively straightforward.
Ahmed, over here. I have a question on also with the iron. Have you looked at the iron content of your piggy soy? And also, have you looked at the amino acid profile? Has that changed dramatically?
Preliminary results, I don't have the iron content. Amino acid profile is also being done as we speak right now. And that's where we're trying to generate enough product to be able to do that analysis, batch analysis. Those are the stages we are at right now. Obviously, for any further approval for consumption, we have to show equivalence in nutrition. So that's the stage we are at currently. But the fact that it's pink is telling you there's iron. We are just doing the analysis right now. I mean, there's more information, but it hasn't been publicly disclosed yet. So.
And another question over here.
Amit, great talk. My question is, like, you share the table where the protein content stayed 35%, and 10% is this new protein. So what is replaced by this 10%?
That's what we'll find out. I think that's the question about we are curious about is, have we changed the amino acid profile? And if we have, is it in a way reducing its quality of the product or not? We'll have to see that. Great question.
Yeah, good talk. And I only have a very simple question. I assume the proteins are expressed exclusively in the seed of soybean. My question here is this. Have you seen any phenotypic, I mean, just, you know what I mean? That's characterization or whatever that is.
Yeah, yeah. Well, so far, a lot of the production has happened in the greenhouse. Not a great place to see any phenotypic differences. That's why we want to do field trials. So far, we have not seen any differences or major differences in that. And I think you had another question, sorry. So that's the first answer.
I mean, the other one is exclusively expressed.
Yes, so the regulatory elements ensure that it's seed-specific expression. Absolutely.
Got another question just over here.
Congratulations, Amin, on all the progress.
Thank you, Jeff.
Two questions, actually, and congratulations on USDA approval. Since this is ultimately for human consumption, I'm wondering what are the FDA and EPA issues growing a human protein in plants? And along the same lines about the channel, how are you going to contain these and not get them mixed in the other parts of the food chain?
Let me answer the first one. So now that we have the RSR from USDA, we will be doing field trials to collect enough information and do all the biochemical analysis that create a dossier for FDA purposes. So we'll go through that process next. Of course, if you look at it, cellular ag, our process, these are some new concepts. And I'm sure you're reading in the newspapers every day that people are expressing casein in different crops. I mean, this is a new category altogether. So we're going to be working with FDA to see what they need, what information they need for safety and equivalency. The channel would be, we'll have to obviously look at that as well, how we're going to grow this and what that business model will look like. Yeah, those are to be determined. There are some conversations happening where we're discussing various sort of models, business models. I can just speculate right now, but there's nothing in concrete to share right now. But those are some good questions, yes. Thank you. Oh.
How about now? Oh, sorry. So one other thing to think about, Amit, is when you start expressing a large amount of a different protein inside a seed and you want to integrate it into agriculture, that seed, actually, the texture, the hardness and quality needs to be the same, or at least as good enough to be handled by the equipment, right? By planting equipment, harvesting, and so on. So have you looked at that with this particular protein? Because like opaque 2, that's part of its failure, right?
Yeah.
Yeah.
Well, we haven't looked at that. Again, same thing. We are starting to scale up the seed production now. I think we are sitting at T4. We've identified 10 events that we are going to scale up and start doing those trials as well. But that's a good point. And I know you used to have a machine to do that, right?
So on this protein sequester iron, have you looked at the long-term effect on environment, iron depletion in the field?
Have we looked at iron depletion in the field? No, we have not, because we are going to embark on these field trials very soon. Actually, we have already started planting this last month as well. So we will be starting to look at all those aspects. That's our next phase. So it's like right off heart of the press or the crushing machine, if you want to call it. This is product. So we're just excited to reach at this point, and now we have the USDA approval to do those trials and evaluations.
Let's thank again Amit for this great presentation. And now we're going to move to our last speaker for this session, and Deepika is going to introduce the speaker for us. Thank you.
Good morning, everyone. It is my pleasure to introduce our next speaker, Dr. Caitlin Hefferon. She teaches microbiology at Cornell University. She did her PhD from Department of Medical Biophysics, University of Toronto. She has published numerous papers, chapters, and review articles, and 3 books. She is also Fulbright Canada Research Chair of Global Food Security and a founder and CTO of Forte Protein. Her research interest includes the use of biotechnology to promote global health. With that, please join me in welcoming Dr. Caitlin Hefferon.
Okay. Hi, everyone. So yes, I've got here the founder of Forte Protein. This is true. I took off CTO. We are actually undergoing a bit of a reorganization of our company. If you go to our website right now, this is what you'll see. It's a lovely video and there's music. You should turn on your sound, and there's a lot of lovely pictures of nature. And definitely we are redefining our relationship with the natural world. As a result, though, of this reorganization, we're in a bit of a stealth mode. So you'll go to our website and what on earth is this? We don't know what we're doing. And so I have to be quite quiet about the actual details of our company today. So I've altered my talk a little bit as a result because of this recent reorganization. And this is what I do most of the time. I'm teaching very large classes of students in microbiology, and it's a lot of fun, but it keeps me very busy. This is my day job. So for today's presentation, I will be talking a bit about the state of the world, need for non-animal source protein. We have had a little bit of conversation about that already with the last two speakers, so I think I can really drive it home a little more with some more information that I can provide. I will talk about molecular farming in general, and I will show you some other super cool work that has nothing to do with Forte Protein, but could be a company on its own, and maybe it should be. And then I'll talk a little bit about my entrepreneurial journey just for a few moments, and this is more for the students and postdocs in this group, because I think you could all do this. There's nothing special that, you know, I did, and I think that this might ring a bell for you to get going and start your own startup. So first, let's just talk about food insecurity. I think we all know this in this crowd, that we have a real problem. I think we reached 8 billion people, what was it, a year and a half ago in the planet. And obviously, we have at least 800 million people who are suffering from hunger. We have about 2.3 billion who are food insecure, and we have hidden hunger. We have people who do not get enough micronutrients. There might be 2 billion or more people in the world right now who are in this kind of situation. And this might be something you may not be aware of. It wasn't really brought up in the last two talks, but the value of our global food system is around 14 trillion, but that's really only about half the story. It's almost double that when you think about the global hidden costs for our food system. And this is things such as negative health outcomes due to eating foods that aren't necessarily the best for us, or the diets that are the best for us, and also the negative environmental effects that are taking place around the planet because of our current agricultural systems. So the price of food is something else we haven't really talked in much detail. I think it was touched upon a little bit with the two other speakers. But we subsidize our food right now, and we're kind of unaware of that most of the time. So a Big Mac, if we went to to McDonald's, would really cost about $13 instead of $5 if we weren't subsidizing our meat, and a pound of hamburger would be about $30. So we really have to do something to transform our food system. We're really slashing prices on things that aren't necessarily the best for us, such as sugar, and where we've got a real unequal balance of how we're providing our food to the global population. And of course, I think most of you are somewhat aware of this topic as well. About 78% of the world's poor are involved in agriculture. So there's an inequality going on. There's amongst the world's population, and who is poor and who is working in agriculture. So it'd be really nice if we could change this up a little bit. The data I'm showing you actually comes from World in Data, I think it is, or World in Data, I think that's the name of it. And we're running out of arable land, so this I think you all know as well. In this graph, I believe the red is grazing. This is land used for livestock, basically, so you can see how much of our land is used for livestock, and then the black part of the dark green part is really for our own cultivation, for directly for our own agriculture. And of course, this should be touched on as well. We are just terrible with greenhouse gas emissions in our agricultural system. So looking at this, the left-hand side, we have methane emissions, and agriculture is way at the top. It's been at the top for a long, long time. So terrible greenhouse gas emissions. On the right-hand side, we have nitrous oxide. That, I think, would be directly through the use of fertilizers. And again, look at how much it's climbed. So it's just way up there, agriculture way above everything else in terms of greenhouse gas emissions. Now, of course, we have been engineering our food for eons, and we talk about molecular biology and genetic engineering, but certainly we have been directly working with our breeding processes for many, really thousands of years to produce the foods and crops that we use today. So on the left-hand side, we're looking at a natural watermelon, this little tiny thing that was, I think, quite bitter to taste. And then we have what we call the artificial watermelon, but the one we eat today that's so sweet and lovely and big and doesn't have too many seeds these days. And then on the right-hand side, we have, I think it's teosinte, I hope I'm saying that right, but it's the original corn that came from Mexico, and so Latin America. And then we have corn of today, and we have so many different varieties, and it's such a major food crop for the world today. So just giving you an idea that genetic engineering, even if it's more of a classical plant breeding, has been around forever. So here is a timeline of cellular agriculture. I thought this would be kind of cool to see. You can see that back in the 1960s, we were starting to work on fermentation technologies, and then in the mid-70s, we started to work on recombinant bacterial DNA technologies. Moving along, we come to the 2013, we have the first beef hamburger that was discussed. So that was the first time we were able to culture meat and make something that resembled a hamburger. And then all the way, you can see things like synthetic vanillin being produced, resveratrol being produced in plants, these sorts of things. And finally, if you look at 2020 at the very end of this timeline, you can see a company, Perfect Day, which uses precision fermentation to make dairy proteins, and they've actually made an ice cream that you can go out and purchase, I believe, and this is using a microbial fermenter to make the dairy protein. So we've really come a long way over time. So precision fermentation, I'll just say a couple of slides or talk about it a little bit because the other two folks I could see were probably not going to, so a little bit more about this. And you can see on the top here we have a cow, and the cow has a rumen. The rumen has a huge diversity of microbes that produce protein at the end of the day. And of course, on the bottom, we have precision fermentation. Well, this is where we have a fermentation tank, and we would have maybe one engineered microbe that we would use, and we know how to grow it under very defined conditions to produce a particular protein. And that protein, may have been a pharmaceutical protein 20, 30 years ago, but now we're thinking actually of food proteins that we can make using this type of technology. So on this chart here, this table, you can see all sorts of different proteins that have been produced using precision fermentation. So Perfect Day is here at the top, and then you can see we have proteins such as egg white proteins being produced, and caseins or cheese proteins, and of course some meat proteins. So all of these can be now made using precision fermentation. As another example of how we're using alternative proteins, we know so much now with our artificial intelligence and the way we're able to use programs such as AlphaFold. We're able to now find what those properties are of plant proteins that could actually mimic or resemble the taste and mouthfeel of some of the animal proteins that we know and love today. So this is a company called Eat Just, and you can see that they've made scrambled eggs that are in fact actually made of mung beans, but you wouldn't be able to tell the difference. I think you can buy this right now. And mayonnaise that actually comes from pea protein. So we're able to re-engineer, just with our knowledge of food technologies, just our standard plant proteins so that we can use them and mimic what we would find in animals. I was talking before about greenhouse gas emissions and the use of land, and this table comes from Good Food Institute, I believe. And it just gives you some idea of where we stand when we're using alternative proteins. So here we have greenhouse gas emissions for products such as the Impossible Burger, or a variety of other things such as cultivated chicken. And you can see the land use as well. So in general, we get a big reduction of greenhouse gas emissions and land use using these sorts of proteins. So it should be said that we can also alter the geopolitics of the planet by using alternative proteins. So different countries may have very limited or small amounts of agriculture. Think of Singapore, for example, this tiny place with a huge population, or Saudi Arabia, which really has very high energy, almost free energy for themselves with their fossil fuels, but they don't really have a way to perform their own agriculture. This could turn things around and perhaps make them the larger food producers than they are today. Because they have access to energy, there is a food energy nexus, so they're able to actually use this to produce different foods in this alternative fashion, different proteins in an alternative fashion. So I think Mulek showed this a figure that's somewhat like this, but it's this alternative protein production landscape. We do talk about three different pillars, which are the mixtures of plants I mentioned before, such as Eat Just. We've talked about cultivated meat in the first lecture. We talked about precision fermentation. I brought it up with you a little bit. And then molecular farming would in fact be a fourth pillar. So we have this way of producing proteins using plants instead of using a microbial fermenter. And so from my point of view, the future of molecular farming looks very, very promising indeed. And you can see here that there are all kinds of players coming into this space. So molecular farming has been around for a while. But look at all these new companies. This is just actually a screenshot from a newsletter called Green Queen. If you're ever interested in following the alt protein space, that might be a good one, a good link to look up, Green Queen. And you can see, at least a dozen, maybe more than a dozen new companies have come along in the last few years, and they're all producing alternative proteins using plants in some fashion or another. And it's really cool to look at all the different ways that this is being done. So molecular farming, actually, I think some of you would know, but some of you may not. It's been around for maybe 25 years, and this was a way for us to produce pharmaceutical proteins. This is the first way we started to look at this, using a plant production system. And it's, as I say, it's been around for a long time, so now we're using it in a different way. And I think this is kind of ironic because originally I come from spent some time in a lab where some of the original work was being done. And I think we thought that we would produce edible vaccines you could eat. And it's kind of come around full circle. So while there are pharmaceuticals made in plants, we can also now make these animal food proteins in plants that we would eat. So it's kind of fun for me to think about this. So just talking about molecular farming in general and how people got into this sort of field in general. Certainly, it was very interesting for folks who were thinking about helping those in low to middle income countries. We could use these sorts of plant-derived vaccines to bypass things such as needles and syringes, refrigeration, possibly some trained medical personnel. They tend to be very low in cost and safe and easier to scale up. So there were a lot of reasons to pursue this field and develop this field more. These figures just come from a colleague at Doctors Without Borders who could, present that making vaccines in plants could enhance the availability to developing countries of pharmaceuticals that they may not necessarily get good access to. And this is an example that you may have read in the news. It was kind of famous at the time, I think around 2015. This was a plant-made vaccine that was produced, it was actually not a vaccine actually, but monoclonal antibodies. It was a cocktail against Ebola. There was an Ebola outbreak around 2015 in Western Africa, and there was a group who were producing monoclonal antibodies. This was used as an experimental medicine to help folks. There were some Americans who had been in Western Africa, came back with Ebola, and so it has some kind of interesting story there. And I like this because I still think that we can use plant-made pharmaceuticals to make an impact on global health. We can either just, completely purify, as you see on the top, a pharmaceutical protein that we may make from a plant, a crop, and provide those to developing countries. Or on the bottom, we don't necessarily have to completely purify the protein. We can even just make a partially purified or crude extract, such as lyophilized tomatoes, that are producing a particular vaccine protein, and then resuspend them at a particular concentration and provide those. Or my favorite of all is just to provide the technology to low and middle income countries so that they can provide pharmaceuticals to address their own local needs, and orphan diseases that we don't really fund here in the West, but perhaps they would really like to have a handle on addressing these concerns in their own countries. So this is some work that I did do. It was right before the pandemic, actually. I interact with a lot of developing countries trying to, work with them, and I find there's a lot of stumbling blocks to do so. But one was the Egyptian government. They were really interested in looking for some way to deal with MERS. MERS, ironically, again, was Middle Eastern respiratory syndrome, and it's a coronavirus, so we hadn't even been hit with COVID-19. And here was a project to work on that dealt with a coronavirus. I was working at University of Toronto for a while at that time. I'm showing you in the diagram that the camels I'm not sure, it's a very high proportion of camels, it might be as high as 90%, actually carry this MERS, and it's endemic in the population. And every once in a while, it passes on to humans, so it can be a real problem, and no one really knows how many folks have died from this. There's outbreaks, it's a respiratory disease. No one is testing why these people seem to contract pneumonia and die, but it's well known in some of these Middle Eastern countries that this sort of thing happens. So I joined forces with a colleague, Dev Sidhu, at University of Toronto, who was actually he was using a phage display mechanism to make antiviral proteins, and he had one for MERS. So we thought we could make this in plants. The picture here of the camels, actually, I took this one. It was somebody's backyard, and people in the Middle East, and you can see all over, actually, if you think of the equator, there is a camel culture. Lots of people have camels, just like we have cats and dogs. So camels are your pets, and they're all over the place. And you think of how many people live here. Several billion people live in this area on the map. Those are old camel places where camels would be walking, these trading routes for camels. And camels are all over here. And these camels can carry MERS, and it can have a 40% fatality rate for humans. So we developed a particular antiviral protein in plants that we could then put in an inhaler, with the idea that if someone came down with this virus, or perhaps their family members could protect themselves in some way. Of course, right around this time, we were hit with COVID. So we had to think quickly, and Dev was able to make a variety of antiviral proteins to not just MERS, but also SARS-CoV-1 and SARS-CoV-2. So we have all three that are functional for each of those viruses and specifically block their replication. That protein I'm showing you there is the replicase, so it's no longer able to replicate in cells. And then Cornell actually shut down pretty much during the COVID pandemic, and we were unable to even go into the lab and do anything. But Sean Chen, who is also a molecular farming guy and at Arizona State, was able to keep going, and he actually had some live COVID of a variety of variants in his lab. So I sent him what I had, and he was able to keep working on this. And he made a plant-based norovirus like particles. So this is norovirus with no DNA in it, made in plants, just the coat protein assembling in plants. We put our antiviral peptide on the surface. This norovirus-like particle can enter cells. It enters epithelial cells. And he continued the work. He has some really exciting data, but it's his story, so I won't tell you any more about this. But that's what was going on during COVID. So I hope I've explained to you that there's lots of things we can do with plants in terms of pharmaceuticals. We can make monoclonal antibodies. We can use them to produce different lectins. We can use them to produce vaccines. There's lots of different things we can do. But I want to talk a little bit more about this other molecular farming approach, which I think you might find interesting. And I think there are a couple of startup companies working on this, and maybe I'll start another one. But this deals with plant viruses. So these are 3 plant viruses that we often use in the field of molecular farming. We have tobacco mosaic virus at the top, potato virus X in the middle, and cowpea mosaic virus on the bottom. And I'm showing you their sort of a graphic of what they look like, and then what they look like under the electron microscope, and then the kind of infection they cause in crops. And so they're easy for us to use, and we have been using plant viruses for eons as well, because they're very manageable and manipulatable in the lab. And so here is example tobacco mosaic virus and all the different types of applications that have been worked on. So there's so many things we can do with these viruses. And I've circled here imaging, diagnostics, and pharmaceuticals, vaccines as what we're talking about right now. So we know the atomic structure of these plant viruses. They're easy to work with. So knowing how they're formed and how they assemble and exactly all the intricate details about them, it's easy for us to use them as either antigen presenting systems or in fact carriers for drug payloads. And this is an example here. This is PVX, and we've had doxorubicin added to this particular, so it's an anti-cancer drug that's been conjugated to the surface of this virus particle. So it's easy enough for us to figure out how to do this sort of thing. And looking at cowpea mosaic virus as an example, there is some work done by the Steinmetz lab, and they were able to show that this cowpea mosaic virus as an empty virus particle can in fact elicit an immune response in multiple ways. So it had some benefits that we were not expecting. So it made us reconsider cancer immunotherapy by using these plant virus nanoparticles as a way to go. And this experiment, I love it because the authors were emailing me and saying, "What do you think about this, Kathleen?" when they were just working on the data, and I just about fell off my chair because I had all of these viruses in the fridge in high quantities, I guess, and just never thought of trying experiments like this. But here we go. So in this case, they've taken cowpea mosaic virus, empty virus particles, just produced it in plants, and had it reassemble, and then they've injected it into the tumors of mice, and they found on the top here you have the empty virus particles, and this is a mouse liver, and they're actually promoting tumor regression. So they're getting rid of the tumor by inserting these virus nanoparticles into the tumor area. On the bottom there we have PBS, so phospholipid saline, and there's no real difference. It's a negative control. So actually using these virus nanoparticles to block tumor progression. So this diagram basically shows what's going on from a plant person point of view. We make the virus particles, we can inject them into a tumor. The tumor contains what we call quiescent neutrophils. So the microenvironment has quiescent neutrophils that become activated in the presence of this of the virus particles. They release cytokines that then attract T cells, and the T cells will lyse and kill up the tumor. This shows you the same thing from more of a medical perspective. So again, inject virus-like particles into the tumor cell. Over time, the quiescent neutrophils become activated, and we end up with a reduction of the tumor. So we can use virus-like particles for all kinds of therapies, either just empty virus particles, or we can add ligands, or we can fill them with payloads, with different cargoes, et cetera, et cetera. And this is just showing you that we can do things such as, turn on cytokines, we can turn on interferon, and we can turn on an immune response in different ways by tweaking these virus-like particles. So just to give you some conclusions about this, they're very easy to manipulate in the lab, and they're biodegradable. They don't last in the body for too long. They're very inexpensive, easy to upscale, safe and efficacious. So now I'll just spend a few minutes talking about my entrepreneurial journey. How did I start this company, which has been running for about 2 years now, Forte Protein? And I had technology in the lab that I've been working on for a while. Cornell sent me actually to rural Botswana for a project, and I was a little bit shocked at what I thought I was seeing in terms of children and protein deficiency. It made me very concerned. We have a lot of people on the planet. We're going to have more on the planet. I noticed a shift in consumer behavior. People are more health conscious or thinking more about getting away from animal source foods. So all of these things together were mulling around in my mind, and I thought Forte Protein might be a fun thing to do. It was at least a way of getting, advancing some technologies that we already had available and maybe doing some good in the world. So very basically, my journey was working in the lab and finding that, I kept getting calls from companies and folks who wanted clones and plasmids and different things that I had, and I wasn't really sure how to use them because I hadn't, they weren't patented, and it was a mix-up, messy with the universities involved. And then actually having two prominent business schools contact me and say, we've read your paper and we want to showcase your technology in a business competition, which I thought was pretty cool. So that got me thinking, well, if they want to showcase my technology in their MIT, maybe I should be starting my own company. This seems like something I should be doing. And then what really got things going was co-writing with a colleague, an op-ed in the Wall Street Journal, and getting some public, something out there in the public. I managed to immediately get an email from an investor, an angel investor, someone who was just a very, very wealthy retired guy who said, I really like this so much, I want to start a company with you. And then that was it, we had to get going. So we started working on a startup, and this was actually in Toronto, of all things. And then COVID-19 hit, and we had a closure of the border between Canada and the States, and it was not possible for me to easily go back and forth, which made it very difficult. So there was we actually closed down our startup because of that, because it just looked like COVID would never end. But then I got going again in New York because I was frustrated, and I found another angel investor through the Good Food Institute. We had something known as speed dating, and he thought this was interesting enough that he wanted to be involved. And then I joined a number of programs. Some exist at Cornell with entrepreneurial work, and some were ones that I just applied for. The Clean Tech Open is all about the environment, but they're very interested in food as well. If we can reduce the environmental impact of food, then yes, they will take you on. And the other one was Indie Bio, which is a SOSV. It's in Manhattan, and they're interested in life technologies. And that's what really got Forte Protein moving forward. And that's all I'll say about this. But thanks so much for listening. That's Cornell, and I'll take some questions.
Questions?
Thank you, Kathleen, for a nice presentation. So I have a you made several examples, gave several examples of viruses used in potential therapeutics. What is the future of this field, given the fact that Medicago, with hundreds of millions of investment, is closed, which did have a product approved for the coronavirus vaccine, and many other hydroponic facilities using the virus technology in the US, Fraunhofer, IBO, have all been closed. So what is the future direction for this field?
So thanks, that's a really good question. So Medicago went through a business change. I don't know if you've spoken to Marcel Dost, I think it was the CEO or of Medicago. So this is a big company in Canada, actually in Quebec City, that made plant-made vaccines for COVID. They put all their ducks, all their eggs in that basket, and then they are owned by a large Japanese multinational, and that multinational just changed direction, and so they closed down. But then I believe they've opened up as another company. They've sort of like a phoenix come back, and there's another company in its stead. I got some LinkedIn invitation, and some of the old folks from Medicago are there. So it's just a business choice. Another one I think that went down is iBio. iBio went down, but when I talked to the iBio folks, and in this case I was talking to them about CDMO and building manufacturing facilities for Forte protein, they also just made a business decision. So they're going in another direction, and they decided to stop the plant work. So I think that just happens, there's just global changes and business decisions made. But at the same time, you see that there are all of these other companies that have cropped up, and they all seem to be moving along, and it's a nice community, actually, molecular farming. Most folks are quite friendly, and I'm finding it a good spot to be in. Most of them come from academic places as well, which is helpful. So we've all sort of known each other, or the labs we've been from for eons as well, yeah.
Very exciting talk, Dr. Heffernan. We are working with collaborators to freezing those lipid nanoparticle for tumor treatment, similar mechanism. But their lipid nanoparticle is highly, thermally unstable at room temperature, like the Pfizer vaccine. So we have a way to freeze them at -20. But it seems to me your plant virus is highly stable, thermal stable, especially this one. You mentioned as CPMV.
Yeah, it seems to be quite stable.
Okay, we should have that.
So that's Nicole Steinmetz's work, and she would be a better person to talk in more detail. But she has really, really interesting results, and she's certainly the forerunner in this field, I would say, with the virus nanoparticles and cancer.
Great.
Yeah.
Also, how's the migration efficiency of this plant virus into solid tumor? For traditional lipid nanoparticle, the migration efficiency is really low. They can only kill the cells on the surface, introduce apoptosis on the surface of tumor. How about the plant virus? Can they get into the...
So it's interesting that certain shapes of viruses seem to work better than others. So we have the rod shape, like TMV, seems to wedge itself into certain types of tumors, and we have a icosahedral virus acts a bit differently. I think all of this work is really ongoing, so those specific questions are good ones that are undergoing research right now.
Seems to me, because the rod one is really different from the spherical.
Yes.
Definitely we should get into this.
Yeah,
the migration.
High aspect ratio, I think is the word they use, yeah.
Thank you.
Yeah.
This is a bit about your entrepreneurial journey. So you mentioned you started the company in Toronto, of all places. I'm from Toronto, and it was kind of almost a passing joke that entrepreneurship is maybe not always happening in Canada. So it made me think about What attracted you to move the company to the States? Maybe a bit of your personal journey. I know COVID was probably playing a role.
Oh yeah, so I was actually on a Fulbright scholarship in Canada at the time when I wrote the op-ed piece for the Wall Street Journal, and the person who contacted me found, I think somewhere on there on the Wall Street Journal article said I was from Toronto, and that's what binged in his mind. And then he was a retired wealthy guy who had run, a food company, had, $600 million in his pocket and was a bit bored. And he was looking for fun things to do, and he thought this looked like a lot of fun. And so we did get started. We were at the point of, had lab space and buying equipment, had folks to work there, old scientists, friends in Toronto. And then it was just a nasty shutdown with the pandemic. First, we waited it out. We waited about 6 months, and there was no end to the pandemic in those days, and the border was shut. And then eventually said, I don't know if I can wait any longer. I think we should just close. And I was very frustrated with that. And then there I was stuck in New York. I'm going to just start this myself. I've had enough of this. So then, fortunately, I found out about the Good Food Institute, and they got me meeting people. This is a DC-based, I guess it's a government institute about alternative proteins. And there's all sorts of resources. So before I knew it, I had someone else who was interested, and he was ready to put down some money. He had originally advertised a $50,000 grant, and when we started talking, he said, oh, I like this so much, I'm going to give you $500,000. And that was what really got it going. And then it wasn't that hard to find other places, such as the SOSV investors who were willing to give this a shot. And the next thing I knew, we were flying along. And that's not to put down Toronto. There's good things going on there even now. It was just this crossing the border thing that was so difficult.
No? Okay. Thank you so much, Caitlin, for a great talk.